go to field. So out of 9 seed lot, 8 of our seed lots are located in the same bay, which means that they are provided with same environmental treatment. And there is very narrow aisle in the nursery, and we use our tripod, and that tripod is modified according to our research need. And at the top of the tripod ball head, we have used this flat wooden structure, and we have tied that wooden structure with the ball head of the tripod with the zip ties, so that it could provide platform for this long telescopic pole to slide back and forth. And this telescopic pole is extendable in size. As you can see, there are 2 knots, and it's extendable into long size, or you can even make it smaller. And at the other end of the telescopic pole is tied with the GoPro camera. This is the zoomed version of how the camera is attached with the telescopic pole. We have used ball head and screw to attach our camera with the telescopic pole. And as you can see in the figure, one of the researchers, they need to get inside the position of 2nd or 3rd stair block. And it depends on height, because my research assistant, she is tall, and if she is to go inside, she will obviously use her shoulder, and me being short, I use my head in order to stabilize this telescopic pole, because it might swing when we are transferring that telescopic pole back and forth. So the reason for doing that is to maintain the height consistency between the seedlings and the GoPro camera. And this is pretty much what does data processing looks like. I'm using AgSoft PhotoScan and ArcGIS for image interpretation. And I got this image from one paper, and this is the whole data pre-processing pipeline. It starts with the image capturing to aligning image, building dense point cloud, and measuring biomass, height, and so on. Okay, so I already measured height on the 15th week from sowing, and I do have combination of 5 seed lot. As you can see, here are 3 seed lot, and here is the combination of 2 seed lot. On x-axis, we do have seed lot, which is improved and unimproved, and on the y-axis, we do have height. So there are the 5 combination of improved and unimproved seed lot, and out of 5 combination, only 1 combination of seed lot shows that unimproved is taller than the improved seed lot, but not significantly taller. But other than that, the other 4 combinations shows that improved seed lots are significantly taller than the unimproved seed lots in the greenhouse. As you can see from this bar plot, that improved seed lot is significantly taller than the unimproved seed lots. So this brings us to the most important question for our research. We do have same lifting specs for the alberta. It's same for improved and unimproved seed lot. And this brings us to raise very important question. Is our nursery practices optimizing or limiting the benefit that we are supposed to receive from the improved seed lots? Because in Alberta, the genetic worth is calculated on the basis of height. But if the nursery are limiting the growth of improved seed lot because they have to meet same specs for improved and unimproved seed lot, so is it even worth it to get improved seed lot to plant, because at the end of the day, we are making similar height for improved and unimproved seed lot. So this question is for the respective stakeholder who are working on that field, and our research is trying to bring out the benefit of raising the improved seed lot, but because of this specs, it seems like we are limiting the benefit that we are supposed to receive from the improved seed lots. And yeah, that's pretty much of my presentation, and I would like to acknowledge following people: Tree Improvement Lab, my supervisor Barb Thomas, Kennedy and Emily for their immense support in the field, Stacy, my lab coordinator, and most importantly, PRT Beaverlodge team, because they are the most friendliest and most helpful people I have ever met in my life. Okay, that's pretty much from my side. Any questions?
Thank you very much for your presentation. In regards to the nursery heights, the nurseries holding back operational seedlot heights, did you look at the ratio of the root collar diameter to the height, and if that was different between the improved lots and the wild lots? And if the root collar diameter was in a proper ratio for a sturdy seedling to be operationally planted in the improved lots that were a bit taller?
Okay, so I just collected the lifted seed lot from the nursery before coming to this conference. So I haven't still measured the root collar diameter for the seedling, because what we thought that if we are measuring root collar diameter, we would definitely do that at the time of lifting, because we did not want it to disrupt the seedling. So at this moment, I don't have answer for that question, but I will definitely get back to you once I have that data.
Hey, thank you, Sarun, for the presentation. It's very close to what we do, so I'm very interested in the results. I just wondered, you're talking about how the nurseries may be holding back the improved seed lots, but do you think there would be any biological trade-offs with letting those improved seed lots grow further when it comes to planting them out in the field?
Okay. So what I have understood is that the improved seed lots, they are supposed to grow taller, because that's how the genetic worth is calculated in Alberta. But as far as from my observation in the field, even you can tell from your naked eyes, even without any data analysis, that the improved seed lot are really growing taller. And also I can tell that their needles are much more scattered as compared to the unimproved seed lot. But there is one thing I noticed about those seed lot. Unimproved seed lots are giving off bud pretty earlier as compared to improved seed lots. So that might be another factor that we need to consider about improved versus unimproved seed lot. And also, in terms of other biological factor, I think definitely the root collar diameter for the improved seed lot might be, as far as for my observation, like it's not, I haven't done any data analysis so far. So it might be slightly thinner as compared to the unimproved ones. So, yeah.
Okay, I think we have time for one more question.
So I wasn't quite clear. Did you use the images to get your heights?
Yeah.
Or, okay, there was no actual measurements with the caliper or anything?
No, I did my ground truthing measurement. So I measured the height on the 15th week, and the result that you saw, so the result that you saw from here, it's from the manual measurement, it's not from the image analysis. So
that's the data that we saw here?
Yeah, the data that you saw here is just from the manual measurement.
Okay, thank you, Sarun, for the perfectly timed presentation.
Thank you so much.
Our next speaker is Keith Jayawickrama. I tried. Welcome, Keith. Let's give him a warm welcome.
Well, good morning. Appreciate being able to talk at this conference. Thanks for the audience. Just going to talk very briefly about two important trials that we put out in the last decade. We've had a long-term interest in realized gain, because ultimately all the numbers from the progenitors are great, but the people who support our program, who use the seed, want to see that's actually growing more trees. So quick rundown on two major species, Western Douglas fir and Western hemlock. And these are our second cycle gain trials. So we have quite a history in gain trials. I'm going to use my own timer here. Going back to the early 1990s, when Brad St. Clair convinced the members of the Northwest Streamfront Cooperative to put these trials in, it was a great move. So those trials are now getting up to age 30. So I'm going to quickly give you a rundown of what we found, so I won't keep you in suspense. Yes, there was a really good gain at age 7 or age 6 from planting for both these species, Douglas fir and hemlock. it actually exceeded our expectations. The gains were high at all sites, and there was some seed lot by site interaction, but that was not a crossover interaction, just increase in scale. So Douglas fir, this one here, we have a very simple design, one spacing, 2 seed lots, elite tested second cycle crosses from the co-op breeding program, tested on 5 sites planted in 2017, and there were more planted later on. These are big square plots of 100 trees, 10 by 10, at 3.3 into 3.3 meters. And so you can see, let's see, our sites that were measured here in this study. So there was interest in having operational conditions. They were not fenced, and we were reminded what happens when you don't have fences around our plantations. One of our sites was pretty much obliterated. The elk pulled the trees out like in a few days. So that was obviously less trees to measure in the future. But on the other hand, some sites, like this site, had very little browse. So animals are unpredictable. We have data at age 7, and so we have height, diameter, and this is something we are very interested in Douglas fir, forking incidents and ramicons. We look at sinuosity. Let's see. So quick rundown on the results of, these are the least square means heights. So you can see these differences. These are largely due to differences in browse. Of course, there's site variability in these sites, but there's browse results in a lot of that. And then we have diameter, survival. So this is the one that I'm going to focus on. So we replanted after the first year, because that would be almost what you do in the operation conditions. So we replanted once. So this is the survival that we're going to track from now on after the first year of replanting. We did some quick significance tests, and there were big site differences for these traits, big significant seed lot differences, and some site by seed lot interactions. So that was on plot means. Now we have individual full SIP crosses with these tests. Therefore, we also ran tests on the individual tree data, and that allows us to do some further levels of testing. So now we have slightly different significance levels, and this is because we have far more degrees of freedom in this test. But these two main upper level effects are staying pretty consistently different. So these are a little bit busy. I'm going to ask you to focus on these green bars, which are our percentage realized gain. So 24, 29, 28%, 35% gain in height for at these different sites. The blue are the elites, and the orange are the woods runs. So here we have blue, the elites taller in each case. So that's height at age 7, diameter at age 7, volume at age 7. This is needle retention. So some small differences in our score of needle retention differences. And then, of course, forking. We have more forking in the elites in all these cases. I'll come up with a suggested explanation why that is happening. But here, yeah, they are forking slightly higher. Ramicons are slightly higher on the elite treatments. Sinuosity is slightly higher score. And slightly more second flushing in the elite treatments. So this is the form of the seed lot by site interaction. There's no crossover interaction, but these differences are bigger or smaller at the different sites. And again, I think this is probably largely attributed to the amount of browse at each site. So if you have a lot of browse, you might not see as much expression of that trait. So similar pattern, different level of difference at the different sites, but always the elites are consistent compared to the woods run. So I guess when I say woods run, I'm using the same word as you'd use wild stand in BC. So comparing predicted gains from the progeny test sites on the single tree plots versus these large plot trials, we were pleased to see that our age 7 realized gain were actually higher than the predicted gains in the progeny test. That's actually pretty rare for us. It's usually the other way around. So we'll take that. That's a good result for us. So 29% height gain, 43% diameter gain, 143% volume gain. So of course these numbers are high now. They will decrease over time as your base comparison goes up. We have individual crosses, and again you can see some variation in the realized gain for the different crosses. They're all good crosses, but they're not identical. So I guess one of the interesting things about our program, differing from the Southeast, is we have still very little use of individual full SIP crosses in reforestation, but I do bring these up to my members and said, hey, why aren't we planting out these individual crosses which we can make? So that's a topic for further discussion. So we had the opportunity to get a drone out there to take some pictures. And so here we have a group and a dog watching with a lot of interest for the drone. Every time the drone went up, it would jump up and bark and run around. So some pictures from the drone. This is our test site. You can see the trees in rows. So I was curious to see if you can see this from the air. And yes, here's our elite crosses. You see bigger crowns, better site coverage versus our wild stand here. If you've tried to take pictures on large plot trials in the woods, you realize it's pretty hard. The trees kind of get in the way. So I was thinking, well, one day we'll have a drone and we can actually go up in the air and look down. So that was one of the reasons for this trial. So here's myself in the woods run lot, and my colleague Sarah Lippo in the elite crosses. So the drone's probably 10 meters up in the air. So I think we're starting to see that actually from the air. So the drone was in the same place, zooming into myself in the woods run versus the elite crosses. So it's pretty exciting. So take home message, as I said, elite seed lots doing consistently well. It had more stem defect. I came up with one possibility is that we measure these defect starting at breast height. Well, the elite trees got there faster. And so they have probably had a year more to express those defects. So maybe that's one reason that's why we have more of that defect. And some significant but non-crossover site by seed lot interaction. So moving on to western hemlock. So the nice thing was all these trials were planted the same year, so using almost the same design. It's kind of unusual opportunity to compare these two. Obviously hemlock, well, not obviously, not everyone works with hemlock. I'll explain that it's a coastal species grown on the more coastal high rainfall areas. One of the big reasons people want to plant hemlock is Swiss needlecast. So these tend to be needlecast impacted sites. So hemlock is planted. We have a single breeding zone from central Oregon coast pretty much to the northwest tip of the continental United States. There were sites planted in 2017 and 2020. I'm talking about the older ones. So there's 4 installations, elite, 18 elite plots, 18 woods run, so fully balanced. Again, this is a good estimate of woods run from 2 lots, which we planted and tagged separately. We have 9 well-tested second-generation full SIP families. So here we don't measure stem defect as much, but height, diameter, And we had height at age 5, so I was able to get an increment from 5 to 7. Crown width is something that one of our partners is interested in. Pictures of our collaborator here, the Stand Management Co-op at the University of Washington, and myself for the Northwest Streamfront Co-op. This is a joint project between these two cooperatives. We are, very much set up for working as associations of companies in Oregon and Washington. So these two cooperatives are running this trial. And I think that's the same tree 3 years later. Again, we had browse. It was dishearteningly high. You're told that, oh, hemlock is not browsed. Well, I guess that's not always the case. We had heavy browse in some cases. This site pretty much escaped browse from the start, and so did this one. So we have height at 2 ages, an increment at from 5 to 7, and survival. Again, you have an elite lot, a woods run or wild stand on the right. Very similar results from Douglas fir, consistent height difference, site differences, strong seed lot, statistically significant differences, some modest interactions. We did the individual tree data, strong, consistent seed lot differences, and also some site by seed lot interactions, big differences of families within seed lots. Again, I'd like you to look at so I'll just explain this. These are the elite in blue, the woods run in orange, realized gain. So 12, 21, 22, 23% height differences. These are the differences in diameter, volume differences, 125, 102, etc. And this is a height increment. So this is interesting to see that that difference is actually growing between elite and woods run. So site interaction, the consistent improvement of the elite, diameter, volume, and height increment. Again, our realized gains are exceeding what we expected at age from the progeny tests, which is great. Differences amongst individual crosses. So I didn't highlight this as much in the past. This is our tallest 5 trees in the hemlock trial. And I guess I'll skip back. So tallest 5 trees in the Douglas fir trial. So I was surprised to see that the hemlock is actually outgrowing the Douglas fir to age 7. The height increment from 5 to 7 is pretty amazing. It's like up to 3 meters of height difference in 2 years, which is pretty incredible. Our percentage gain decreases from 3 to 5 to 7. The absolute gain in terms of centimeters is increasing over those 3 measurements. Yeah, so similar results as I said. Elite is doing very well. The gains are higher than predicted. The elite has a higher crown width, and the relative gain decreased from age 3 to 7. Absolute gain increased continuously during that same period. So I'll acknowledge that none of this would happen without our long-term members who have supported this research for decades now. So, and with that, I'll stop and take questions.
Okay, so questions?
Keith, did you observe a differential level of browse between improved and unimproved, or the Woods Run material? Or?
I don't think we've observed that.
Yeah, if Dr. Sally John was in the audience, she would ask you about age-age correlation. And since you're realizing gains greater than what was predicted, is that it's going to prompt you to revisit your predicted gain for operational seed lots and attaching higher values to them?
Okay, so I think, Brian, you're asking, do we predict what our gains are at rotation?
Yeah.
So we haven't really taken that step. We are giving them gains at the age that it was measured, and then if they ask us, well, as they invariably do, what is it at rotation, we give them our best guess. We are hoping to use our realized gain trials, which are older, to try and narrow that, get a better answer. So I guess, since we haven't really given, like you do in BC, this really strong, quantitative numbers of this is what you're going to get, because we don't really have to walk it back because we haven't taken that leap yet. Does that answer your question?
Thanks, Keith, for your presentation. I just wanted to share to you and to the group that we recently published age 6 results from our realized gain trials at Loblolly Pine in the southeast, and finding similar things that, when you compare realized gain trials at a young age to progeny test trials at a young age, you get similar results, except for whenever someone rides a four-wheeler through the test, or deer come and eat the test. So I just wanted to share that with the group.
I think there was a question on the staircase.
The difference that you found in just the average height between hemlock and Doug fir is quite interesting. I was wondering if, just in general, the hemlock gain sites were planted on more productive areas than the Doug fir, or if it was relatively comparable.
That's a good question, John. I guess there were representative sites. We may have lucked out that the hemlock were, well, partly those two sites were not browsed. Maybe that's pushing the gain numbers up. It was surprising, and I think it'll be surprising to many. And I'd be very interested to see if that continues through to the future, because Douglas fir is the preferred species, but maybe we are underestimating hemlock.
I have a question, actually. As a student, maybe this might be obvious for them, but so you had 20% increase in height and 38 diameter and 106 in volume. I just wondered how you calculate volume.
It's very simple. It's diameter squared times height. Okay. Because with trees that young, you don't have a proper volume equation. And even if you do, they won't. Most of the trees will be not. They will not even give you a number because they're not set up for trees that small.
Okay, thank you. Let's thank Keith. So our final presenter of this morning is Adam Gilewski. Okay, so let's welcome Adam.
All right, that's fine. Okay, so my talk is Physiological and Transcriptomic Responses to Drought and Ponderosa Pine. This was a collaborative effort with Marie Vance, who I kind of piggybacked off of her experiment. That was run here in Vernon. Yeah, so there's a new breeding program that Marie Vance has been starting for the last couple of years, and that's to address increasing demand in seed for Ponderosa pine, and that's mainly due to climate change. So here you can see in the left sort of map the current distribution of Ponderosa pine in BC. This is a very small distribution relative to its wide range throughout the Western North America that goes all the way down to Mexico. And then on the figure on the right, you can see that there's going to be a lot of expansion, predicted expansion of the species into the interior of BC. If I continue and zoom into this area, you can see in red coloration, there's regions that are going to have decline. So that's most of the current distribution that's found in BC. So these are the hottest and driest forests that we have. There's no more drought-tolerant tree species in BC to replace it here. So we're expecting to maybe potentially lose those forests to grasslands. So there's just generally interest in drought tolerance for this species. So research goals, we wanted to identify if there's any potential trade-offs between growth and drought tolerance. So we want to know if we're going to select for growth, are we going to compromise drought tolerance, right? And then which traits might be relevant and might help explain variation in drought tolerance between the families that we're testing. And then there's very little genomic resources available for Ponderosa pine, particularly for drought-related things. So we use RNA sequencing to generate a reference transcriptome, and then that reference transcriptome was used to identify genes relevant to drought tolerance, or just drought responses, sorry. So the main experiment here was the raised beds trial that was run by Marie. This included 22 half-sibling families from BC's only A-class orchard. So that's the one that we visited yesterday, the Belly Seaward Orchard. This experiment was also run at the Kalamalka Research Station that we visited first before that. So this experiment included 10 beds. These were split between drought and control treatments. And the experiment was run for 2 summers in 2020 and 2021. So the drought treatment was applied by installing rain exclusion roofs over each of the beds, including the controls. And then only the controls were drip irrigated once a week throughout each of the summers. And then the roofs were uninstalled at the end of the summer to allow regular precipitation to enter the beds again between years. So these figures show the dry down within the beds, so just soil. So we have soil volumetric moisture content on the y-axis, and then months on the x-axis. This just shows the dry down of the drought beds relative to the control beds. So drought is in red, control is in blue. And you can see that the drought beds have lower soil water content by about mid-July in 2020. So that was kind of late in the season, so the roofs were installed earlier in the second year, and then we can see that we get more differentiation between the treatments in about mid-June. So traits measured, a lot of traits were measured. I'm just going to focus on 3 here that were sort of the most relevant. So height measurements were taken throughout 2020 and 2021, and then we also measured water potential as well as integrated water sufficiency at the end of September in 2020. So there are some concerns whether there was sufficient dry down, especially in 2020, to actually provide stress to these trees. But we do see that for water potential, there does seem to be quite a bit of differentiation between the 2 treatments. So this specifically is a histogram. And over here we have the controls that have generally water potential above -2 megapascals, while the drought treatment generally has water potential below -2 megapascals. And there's sort of a soft border here at -2, which lines up nicely with literature that shows that Ponderosa pine tends to close its stomata at pretty much -2 megapascals. This is likely due to xylem cavitation that occurs a little bit below this point. So this figure just shows kind of average height growth over the entire experiment for both years and both treatments. So in 2020, oops, that's fine. Here specifically, there's no real differences in treatment between the 2, and that's because height growth occurred before the drought treatment really took any effect, before the roofs were even installed, really. While for 2021, we do see clear differentiation with drought having much lower height growth. But most of this growth also, once again, occurred before treatment really took any effect. So most of these effects are from the previous year's drought. And then just heritability correlations, or sorry, not just heritability. In 2020, about 0.17, and then in 2021, had to be calculated separately because there's treatment effects, and it hovers at about 0.1. So going to trade-offs now. This first figure I'm going to show just has height growth in 2020, which occurred sort of before the drought treatment, so we kind of treat it as a control, and then height growth in 2021, which occurred after drought treatment. And as you can see that individuals that generally tended to grow well in 2020 before treatment still continue to grow well after the drought treatment. So that kind of suggests maybe no trade-off there. So then I also calculated genetic correlations between the 2 years, so 2020 and 2021. I get a pretty similar value to the correlation here, 0.45 and 0.49. But then if I do control versus drought beds within 2021, it's not as clear of a story here. We get a much larger genetic correlation, 0.75, but also comes with a very large standard error. So at most, maybe we can say that it's not negative. So that's, sorry, I should go back. So that's kind of reassuring. It means we can probably select for growth or drought tolerance and not compromise the other one. So then I wanted to look at other traits to see how they relate to drought tolerance in this species. So starting here with integrated water use efficiency on the x-axis, height growth 2021 on the y-axis. We see that for both treatments, there's generally negative correlations between these two traits. So that sort of just means that the individuals that grew better in both treatments also had lower water use efficiency. So they may have been spending more water or just somehow maintaining larger carbon dioxide levels within their needles. Next figure here has water potential on the x-axis instead. Height growth is still on the y. And here there's only a positive correlation between water potential and height growth, but not in the controls, which makes sense. Water was only limiting in the drought treatment, right? So then just overall, this is a little bit strange, especially for the water use efficiency. It's not something you typically see. So it suggests that there might be a drought avoidance strategy, is sort of our current hypothesis. So kind of just to recap, there seems to be fast-growing individuals that have low water use efficiency, so they're spending more water, yet somehow they have more water afterwards. So this might be due to something like expanded root system, or maybe really tight stomatal regulation. So we couldn't really get to the roots in the raised beds, so I did these little hydroponic solution experiments where I'm growing 1-week-old seedlings here. And these hydroponic solutions contain polyethylene glycol, so it's a solute, it lowers the water potential, but it's too big to get through the membrane, so it doesn't interfere in the way that like salt would. Yeah, so I could adjust the osmotic potential by changing the concentration of polyethylene glycol. So here, very basic figure, I just have the treatment, 9 and 17% PEG, which are equivalent to about negative 0.13 megapascals, negative 0.5 megapascals, and the control is just 0% PEG. And what we found surprising is these treatments actually stimulated root growth rather than inhibiting it. So it kind of just goes to show how drought tolerant the species is. And it's also surprising because similar studies in Norway spruce and Scots pine found that 17% PEG almost always inhibited root growth. So, okay. Okay, oh, thanks. That works pretty well. Okay, so I raised the concentration to 22% PEG to hopefully get some inhibition. Maybe it's the antenna. It's a little bit, sorry about that. So we do see some inhibition in some families here, but at the same time, there's some families that seem to have absolutely no inhibition, especially 1007 here and a few others. But these aren't necessarily the best families because we still have other families that have similar root growth in the 22% PEG concentration, but in the controls they have higher growth, so that's probably preferable. So moving on to transcriptomics, we did RNA sequencing. So we sequenced 10 families from the raised beds for needles specifically, and then additional sequencing from buds and stems from the raised beds for 2 families. That was in 2021, and then 1 family whole seedlings from that hydroponic experiment. So these included obviously drought and control treatments, and using these data, I assembled a transcriptome, and I got a pretty high BUSCO score, so that's benchmarking universal single copy ortholog, so it's just kind of a standard measure for gene model quality. But there's other measures too that I don't want to show you a whole bunch of them. So using that transcriptome, I quantified gene expression. So I identified nearly 10,000 genes that are differentially expressed between the 2 treatments. So this heat map shows those genes. Specifically, columns, so up and down, are individual samples that were sequenced. And then rows are individual genes. There's too many genes to show here, so they've been compressed. And the Z-score is just sort of a centered mean of the expression values. So red indicates higher expression, and blue indicates lower expression. And then you have the 2 treatments, so controls in blue, droughts in yellow. And you can see that the 2 treatments clustered separately pretty nicely here, with the exception of 1 drought individual that's among the control individuals. So we have genes that are downregulated in drought. So I have blue coloration here, lower expression, red coloration here, so higher expression. And then vice versa, you have red coloration here for genes that are upregulated under drought. So just kind of functional terms based off of enriched gene ontology, you get the usual suspects, response to water deprivation, response to cold, which has similar signaling pathways to drought, and response to ABA, which is probably the most well-known drought signaling hormone. And then I also get things like regulation of apoptosis, which kind of indicates that most of this higher expression is due to more kind of extreme stress. This was also, I found kind of similar results from another analysis I'm not going to show today. So another thing to note here is that there seems to be an intermediate group within the drought treatment over here. So you can see that they have more intermediate expression of these differentially expressed genes. So this blue is less intense, this red is less intense, right? So if I add some physiological traits here, so I have water potential and height growth. So green indicates higher values, magenta indicates lower values for both of these. We see that control treatment generally has green, obviously. Drought generally has magenta. And then intermediate group, once again, kind of has intermediate coloration closer to white. And then just finally, I'm going to add family here just to show that there's specific families. You can't see it super well. But like this orange one here, which is 1010, lies almost entirely within the intermediate group. There's also 1016 in green, which is entirely here. And then there's 2004 that has 2 or 3 individuals here and the 1 individual that clustered among the well-watered individuals. Sorry, forgot to say one thing. So within this heat map, I can only kind of see genes that are upregulated in the severely stressed group. So I wanted to see if there's anything that's upregulated specifically in this intermediate stress group. So that's what this next figure is going to be. So most of the genes that were downregulated in this intermediate group, which is signified by this blue bar here, or so when it's blue, it's intermediate. When it's red, it's everything else, including the well-watered and the severely stressed drought group. So these ones that have downregulation, most of these were just upregulated in the well-watered, so I'm going to ignore those, at least for now. This is very preliminary. So I'm just focusing down here instead. You can see that there's much more intense red coloration compared to the severely stressed drought group here, while the well-watered has very low expression. So these genes have some potential to be functionally related to why this group might be intermediately stressed rather than severely stressed, maybe. So the most, by far the most significant gene compared to everything else out of these 59 genes is a late embryogenesis abundant protein. So those are very well known to be associated with drought. Especially why they're embryogenesis abundant is because seeds desiccate a lot, so they have to have those proteins in order to survive in that state. And then also the most enriched functional term is positive regulation of ABA signaling. So one of those genes was Zerico that promotes more biosynthesis of ABA as well as sensitivity to ABA. And here are the 3 other genes within that group are all CAR-like genes. So these are genes, I'm not going to go into details here, but essentially they transport the primary ABA receptors to the membrane where they have increased sensitivity, and they're also closer to the ion channels that are responsible for stomatoclosure. So that's like one of the main ABA responses. So increasing sensitivity may allow these plants to more quickly close their stomata in response to drought during the day, for example. So usually this occurs in a calcium-dependent manner where it's released from the nucleus, but there's also cases where if the expression is high enough, then it'll escape sort of the nucleus just by being highly expressed. So in summary, we didn't find any evidence of trade-offs between growth and drought. Successful individuals conserved less water, yet somehow still had more water available to them. We generated a highly complete transcriptome assembly for Ponderosa pine, and we identified genes within that transcriptome that were associated with drought responses and physiological traits. So just acknowledgements, Marie Vance for allowing me to work on this project and allowing me to piggyback off of her experiment, Jim Matson, my supervisor, Tristan Huber, who helped with trait measurements, and Jared Columbus, who provided seeds for the PEC trials. Thank you.
Thank you, Adam. Do we have any questions?
Great talk. Thank you. That was very interesting. You had on slide 17, there was that one family 2004, which is it's drought, but it's on the left side with the controls.
2004, yeah.
Yeah, that yellow, yeah. Did you look more closely, like why that was acting more as if it was, I think that was with the controls than as a drought?
Yeah, it just
why the
has
gene
expression
expression was
that's
like
more
that?
similar to, like, based off the clustering. So if you change the clustering method every single time, the clustering is going to be slightly different, but I consistently use Ward's method, and this is just, actually no, I consistently got this particular individual always clustered separately, even though it's in the drought treatment.
Are you going to look at what's going on with that one in particular or not?
I need replicates to do that, right?
Yeah.
Can't just do
Thank
that
you.
with one individual.
Thanks, Adam. Very nice talk. I'm wondering if you had a chance to compare your transcriptional responses with the responses of other conifers. In particular, lodgepole pine has had a similar look at gene expression under drought and well watered. I'm just curious how much overlap there is.
So the difficult part with that is sort of just lack of annotation, lack of possibilities for comparison. That would require another entire analysis where I identify orthologs and such in order to make that comparison. And that's something I actually really want to do, is maybe try taking all the drought experiments for conifers and trying to do kind of consensus study. Might be cool. Thanks.
Another question?
Okay, so you did this over multiple years, a great presentation also. And I'm wondering if you noticed any infection in any of your trees and if that had anything to do with drought. Just like purely observational.
I don't think we did. Right,
That's
Marie?
very lucky, actually, too. That's awesome. Yeah.
Marie might know better.
Yeah.
Did we see any infection?
Yeah, like, I mean,
think so.
they're isolated somewhat, but like they're also outside, so I don't know, like, is that common or? That's awesome. Yeah, okay. Just wondering.
Okay, thanks. Sure. So just one thing I want to mention is based off that other kind of analysis I did see that successful individuals tend to have higher terpenoid production. So I'm wondering if that might be related to some things that I found, after drought, more pest and infections occurring.
So I had a question about it. Just maybe correct me if I'm misinterpreting this, but you found stimulated root growth at 17% PEG and inhibited root growth at 22%. Is that accurate?
But at 22%, it depended on the family. So for some families, there was no inhibition. I can show it again. Oh, sorry, wrong direction. There we go. So there's the individuals here that had inhibition, right? Control is much higher than the 22% PEG. But then over here, there's some individuals where like no significance at all, right?
I was just wondering what osmotic potential that 22% corresponded to, because it seems like that would still be relatively moderate drought condition.
Yeah, sorry, I forgot to mention that. That's -1 megapascals.
Okay.
Yeah.
Okay, I have one question, or maybe just a comment, but do you think that for your water use efficiency versus height that you presented, do you think there could be anything associated with hydraulic efficiency versus cavitation risk?
Hydraulic efficiency versus... could you repeat the question? Maybe I don't understand.
So you have, here you have trees with higher water use efficiency and lower height. I'm just wondering if there could be any trade-off between the hydraulic efficiency and risk of cavitation, maybe.
Right, so yeah, other experiments have found something similar, or provenance trials specifically. So trees from sort of drier locations tend to have lower growth and higher water use efficiency. But they also didn't find any differences. So yeah, there might be less risk there. Maybe that's why those trees from those particular climates have that higher water use efficiency, is to avoid hydraulic risk and like cavitation. But at the same time, when they actually looked at, say, risk, sorry. They did look at vulnerability of the xylem itself, though, and they didn't find any differences there. But that's not maybe related.
Okay, thank you. I don't think we have time for another question. Sorry. Let's thank Adam. We have one more session happening in the other room, and after that we'll break for lunch. Thanks everyone for being here this morning. Yep.